How does apoptosis affect brain cells after a stroke? Imagine a city where some buildings suddenly lose power and start to decay. In the brain after a stroke, a similar process happens at the cellular level. Apoptosis is a natural, controlled way cells die, but after a stroke, it can contribute to significant brain damage. When blood flow stops suddenly, as in an ischemic stroke, brain cells in the affected area are starved of oxygen and nutrients. The core area of damage experiences rapid cell death, mainly through necrosis, which is an uncontrolled form of cell destruction. But surrounding this core is the penumbra, a zone where cells are injured but still alive. Here, apoptosis plays a major role in cell death that happens hours or even days after the stroke. This delayed process offers a window for treatments that could protect neurons and limit damage. The molecular signals that trigger apoptosis after a stroke are complex. They include an overproduction of harmful free radicals that cause oxidative stress, an overload of calcium inside cells that disrupts normal functions, and excessive releases of neurotransmitters that overstimulate neurons. These events activate both intrinsic pathways, involving the mitochondria, and extrinsic pathways, involving cell surface signals. A key player is a protein called p53, which responds to DNA damage and activates genes that promote cell death. This leads to mitochondrial dysfunction, activation of enzymes called caspases, and the systematic breakdown of DNA and cellular components. The process results in the formation of small, membrane-bound fragments called apoptotic bodies, which are then cleaned up by immune cells without causing inflammation. Apoptosis is not limited to neurons. Supporting cells like astrocytes can also undergo programmed cell death after a stroke. When these cells die, they impair the brain's ability to repair and maintain itself, worsening the overall damage. In hemorrhagic strokes, where bleeding occurs in the brain, apoptosis also increases in the area around the bleed, contributing to tissue loss. Studies show that markers of apoptosis rise within the first day after bleeding, and experimental treatments that inhibit apoptosis have shown promise in reducing brain injury in animal models. Understanding apoptosis in stroke is crucial for developing neuroprotective strategies. By targeting the pathways that lead to cell death, researchers hope to save neurons in the penumbra and improve recovery. Some experimental therapies focus on blocking caspases or modulating p53 activity to prevent unnecessary cell death. While these approaches are still under investigation, they highlight the importance of controlling apoptosis to preserve brain function and reduce long-term disability. In summary, apoptosis after a stroke is a carefully regulated process that contributes to the delayed loss of brain cells, especially in the surrounding tissue that is at risk. It is driven by molecular signals related to oxygen deprivation, oxidative stress, and calcium overload. Targeting these pathways offers a promising avenue for neuroprotection, helping to protect brain cells and improve outcomes for stroke patients. Always remember, if you or someone you know experiences a stroke, seeking immediate medical attention is vital. Advances in understanding cell death pathways continue to shape future treatments, but personalized care from healthcare professionals remains essential.